genome maintenance is very important to an organism. A somatic cell can be cloned and become an individual, like Dolly the sheep. That's because the somatic cell contains essentially the same genome as the germline. However, this genome constancy rule is not always the case in nature. Over 130 years ago, the great cell biologist Theodore Boveri found in the horse parasitic nematode Parascus, the chromosomes were broken and DNA pieces were lost during mitosis, as shown in this original drawing. This resulted in distinct germline and somatic genome. This is known as programmed DNA elimination. Now, as we all know, such a dramatic change to the genome can often cause issues to an organism. Well-known examples are in cancer cells, where the genome is often damaged, DNA rearranged, or lost. Yet, in organisms with DNA elimination, they are perfectly fine. Actually, this is part of their normal life. Over the years, many animals were found to slow away their genomic DNA. You can see this happens in invertebrates and vertebrates, over 100 species in the tree of life, and more are to be discovered. Let me show you what DNA elimination looks like. This is a 4-cell embryo with the bottom 2 cells undergoing DNA elimination mitosis. The blue parts are the retained chromosome going to the daughter cells, while the red part is the DNA that will be thrown away from the genome. This image is from the parasitic nematode Ascaris. Now, Ascaris infects pigs and humans. On the left is an image showing a little girl in Paraguay with Ascaris infection in 1980s. The right image shows the worms extracted from the poor little girl's belly after deworming. Now, if you think this was a long time ago and it's not a problem anymore, think again. These images were taken during a field trip a couple years ago in Ohio, where we collected Ascaris samples from a pig slaughterhouse. I can tell you Ascaris infection in pigs is still very common even in the United States. So how do we study DNA elimination? This is the cell lineage showing where DNA elimination occurs. Red are the germ cells that remains intact, while blue are the somatic cells with reduced genome. The 5 cells labeled in yellow with red dots are cells undergoing independent DNA elimination. Now first, we need to figure out what sequences are thrown away. To do that, we sequenced and compared the germline testis and the somatic intestine genome. This was done using Illumina high-throughput sequencing and lots of computation and data analysis using supercomputers. What we found was astonishing. In addition to many repetitive sequences, we found 1,000 genes were thrown away. These genes are only needed in the germline cells. The organism is using DNA elimination to silence their expression in the somatic cells by permanently remove them from the somatic genome. This is not only the case in Ascaris. In fact, when we look at eliminated genes in closely related worms, the horse and dog parasites, we found that 1/3 of them are conserved and they are highly expressed during the spermatogenesis of these worms. This reinforces the idea that DNA elimination is a way to silencing these germline genes in the somatic cells. Our recent study showed that all Ascaris chromosome ends are removed during DNA elimination. This is a way to remodel all germline chromosome ends. In addition to that, the break also occurs in the middle of the chromosome, which leads to the increased number of chromosomes in the somatic cells. Now, using EM, we were able to determine that the eliminated DNA is in a double membrane structure that eventually becomes an autophagosome, where the DNA is recycled. So to summarize, we know that the chromosomes are fragmented into many pieces. Some of the DNA will go to the daughter cells, while other DNA is going to go to the recycle bin. The lost sequences contain many germline-specific genes. That's how they are silenced in the somatic cell, as they do not exist anymore in the somatic genome. My lab is focused on the molecular mechanism of how DNA elimination occurs. We are trying to address questions such as, how does the cell recognize the break sites? How are the breaks made? And how are the breaks repaired? We are using a variety of modern molecular tools to answer these questions. My lab is also using other systems to carry out a comparative study on DNA elimination. One of them is a freshwater copepod called MicNite. It's a crustacean that has a genome 3 times bigger than the human genome, and it throws away over 80% of the DNA. Our research is important as we are trying to study the basic biology of a parasite that infects close to 1 billion people, mostly children. This is also important for veterinary animals such as pigs and horses. The genomic data that we have generated is already being used in many studies to fight the disease. Our study on the molecular mechanism of DNA break and repair, RNA transcription, DNA replication, and the 3D genome organization may also provide insights into fundamental biological processes that may be relevant to human disease, such as cancer and other chromosomal disorders. Finally, DNA elimination is genome editing at a large scale. If we can figure out the molecular mechanism of how this works, maybe it will lead to potential tools development that may be used to manipulate this genome at large scales. As you can see, DNA elimination can impact every chromosomes, both in the middle and end of the chromosomes. I would like to thank everybody in our team working extremely hard during the pandemic. I would also like to thank NIH for funding. If you want to learn more about our work on DNA elimination, please visit our website. That's DNA elimination in one word, .utk.edu. Thank you.